Good morning, students. Today we start a new chapter. This chapter is being named as Biomolecules. This lecture is presented by me, Pranit Ghatak, your chemistry teacher. So, in this class, we're going to discuss about the biomolecules. This chapter is being deeply reduced as vitamin part as well as the structure of protein is almost removed from the syllabus. We are going according to the further reduced syllabus that is provided by the council itself. Okay. So in this class, we're going to discuss about the some ins and outs of biomolecules. This is a relatively small chapter and it has some relation with the biology as well. You may have already known it as all. Okay, so let's get started. So the first thing, what is monosaccharide? Okay, so in the case of biomolecules, biomolecules are basically uh, that kind of molecules that having carbon, hydrogen and oxygen itself. Okay, and biomolecules can be divided into three parts. Biomolecules can be divided into three parts, that is carbohydrate, lipid and protein. Okay, carbohydrate, lipid or fat and protein. Okay, so in our syllabus we have only two things, that is carbohydrate number one and number two is protein. Lipid or fat is not in our syllabus. Okay, so we will discuss only the two things, that is carbohydrate and proteins. Okay, so in the case of carbohydrate, this carbohydrate can also be uh, distinguished or classified in three types. Mainly this carbohydrate can be classified in three types, that is monosaccharide, oligosaccharide and polysaccharide. Okay, so this carbohydrate can also be classified into three types, that is monosaccharide, oligosaccharide and polysaccharide. What happened if after the hydrolyzation, if the carbohydrate doesn't decompose, or if we can say it as that, if a carbohydrate cannot be hydrolyzed further, then it will be called as monosaccharide, like glucose or fructose. Okay, so any carbohydrate that cannot be hydrolyzed further is being called as monosaccharide. Okay, and like glucose and fructose, if the carbohydrate can be hydrolyzed and it will form carbon number 2 to 10, then it will be called as oligosaccharide. Okay, and after the hydrolyzation, if we get a monomer of more than 10, then that carbohydrate will be called as polysaccharide. Okay, so if the carbohydrate doesn't hydrolyze at all and it will give no further product, then it will be called as monosaccharide. If it gives a product of 2 to 10 after the hydrolyzation, it will be called as oligosaccharide. If more than the product or is more than 10 after the hydrolyzation, it will be called as polysaccharide. Okay, like polysaccharides are starch. Okay, and then it comes to amylose and amylopectin. So what are they? They are mainly, one amylose is water soluble and amylopectin is water insoluble, and this is their respective components as well. And one more thing is cellulose. So what is cellulose? Cellulose is basically a biomolecule that carbohydrate. It is one type of carbohydrate and it has 10 polysaccharide of beta minus D glucose and joined by C1 to C4 glycosaccharide linkage. Okay, and then it comes to glycogen. And what is glycogen? Glycogen is basically highly bunched polymer of alpha D glucose and found in liver and muscles. Okay, so glycogen is very much important. Okay, so it is a highly bunched polymer of alpha D glucose and it is being found in liver and muscles. And in this chart we will be discussing mainly the definitions and what is it. Okay, and in your book you will find the reactions and the structure as well. I draw some structure here so that you get a clear idea on that as well. Okay, so what is reducing sugar? Remember, in the case of reducing sugar, if aldehyde and ketonic group, that is the CO and the CHO, if these two groups are free in any molecule, then it will be a reducing sugar. It can reduce the failing solution. Okay, so in the case of maltose and lactose, what happened? They have their aldehydic and ketonic group, that is the CO and CHO group free. So that is why it will reduce the filling solution. So the maltose and lactose will be called as reducing sugar. But where in the case of non-reducing sugar, the aldehydic and ketonic, that is CO and CHO group, is not free. So that is why it cannot reduce the filling solution, not all in solution either. So it will be called as non-reducing sugar. Non-reducing sugar are sucrose. Okay. And the next topic comes, that is anomer. So what is anomer? Basically anomer are the two cyclic hemiacetal form of glucose differed only in configuration by hydroxy group is being called as anomer, and the C1 at the point, the carbon point is C1, and the this carbon is being called as anomeric carbon. Okay, let me explain it to further. Okay, so what happened, um, so this is the structure of glucose. Okay, that is 1 2 sorry 1 2 3 4 5 and 6. Okay, this is CH2OH, this 6 number carbon. Okay, so what happened in this carbon? What happened, this OH, this oxygen will do a bond. This oxygen means, suppose let me think, this is the oxygen and this oxygen do a bond to here. Okay, so that it will go minus. So right now it will form a product like, and here it will do like this only. Okay, or we can write it in that way as well. OH and H. So this is the structure of the anomer. Okay, and here this is the carbon number 1, this is carbon number 2, 3, 4, 5, and this is 6 here. So this is a cyclic hemiacetal form of glucose. This is called glucose. This is glucose, and after this is a non-cyclic form. This is a non-cyclic form, or acyclic form, we can say acyclic form, but this is a cyclic form. This is a cyclic form. Okay, so in the cyclic form, what we can see here itself, there is hydrogen is being left and this is being right. Okay, and which is being right. So in any other, uh, this is called a anomeric form or cyclic form of glucose. In any other structure, this OH can also be happen in here, and O can be there, and hydrogen can be there as well. Okay, so let me draw it another further time. What will happen right now? So this is the form of anomization or cyclization of glucose, and it's taken very much time he didn't even realize that at all right now. Okay. Okay. So what happened if we do similar things right now? 1, 2, 3, 4, 5, 6. That is CH2OH. Okay. So if it is doing a bond similar like that one, but here this H can be there and this OH can be in the left hand side. Okay, so these two things can also be there. Okay, so depending on the H and relative position of OH and H, these two are different. Okay, but although they are structurally almost same. Okay, just the positions are different, so they are being called as anomers. So both of these are being called as anomers to each other, anomers to each other, anomers. Okay, anomers to each other, and this C1, the carbon 1, is being called the anomeric carbon, and this C1 is being called, this C1 is being called as, this C1 is being called as anomeric carbon, anomeric carbon. Okay, anomeric carbon. I hope you have understood this part. Okay, and what is inverse sugar? So what happens when we know that sucrose is being a disaccharide? It will hydrolyze and it will give glucose and fructose. As the glucose, sucrose is dextrorotatory and it is disaccharide. When it hydrolyzes, it will give glucose and fructose. And the fructose is levorotatory and glucose is dextrorotatory. But the lever rotation of fructose is so higher that the mixture gets levorotatory. Okay, so that is why the sucrose, although it is a dextrorotatory, after hydrolysis, it will give fructose and glucose. The fructose is levorotatory and it has a higher amount of lever rotations due to why the mixture of fructose and glucose become levorotatory. So that is why after the hydrolysis, the dextrorotatory sucrose become levorotatory fructose. Okay, about levorotatory fructose and glucose. So that is why a change in the rotation is being occurring. That is from dextrorotatory to levorotatory. So that is being called as invert sugar. Okay. And glycoside linkage, the linkage is between two monosaccharides being called as glycoside linkage. Okay, the importance of carbohydrate, it is very much important, we know that as well, because it is a major product of our food and we need to store some energy through the carbohydrate as well. And the next topic is the amino acid. As we know, the protein is a polymer. Protein is a polymeric form. Okay, protein are the proteins are the polymeric form, So what are the monomers of protein? Mainly monomers of protein are being called monomers of protein. Monomers of protein are being called as amino acid. Monomers of protein is being called as amino acid. So amino acid have a general structure. What is that mainly? So CH R sorry RCH NH2 and COOH. This is being called as, this is the general structure of amino acid. So here you can see this is the acidic part and this is the amine part. This is called amine part and this is called the acid part. So that is why it is being called as amino acid. Okay, as here NH2 as well as COOH, both of the groups are present in a single compound, so that is being called as amino acid. Okay, this NH2 parts called amino and from COOH is being called as acid. So amino acid is being there. Okay, and what is deuterion? So suppose in a balanced pH, what happened? The COOH will lose one hydrogen. The acidic part of the amino acid will lose one hydrogen and it will form COO minus, and that hydrogen is being captured by NH2. There's a basic part and it will form NH3 plus. Okay, so here zwitter means double. Okay, zwitter means double or two. So two zwitter ions, two ion. Zwitter ion means two ions. So that is a negative ion and positive ion. So here in the compound we have a negative ion and a positive ion. So that is why it is being called, in the compound we have two ions, that is one positive and one negative. So that is why it is being called as Zwitter ionic structure or Zwitter ion, where the two ions are forming and one forms COOH, where the COOH group is being donating one hydrogen and it will become COO minus, and this hydrogen and any other hydrogen is being captured by or accepted by the NH2 plus, that is amine group, and it will form NH3 plus. Okay, so in this way they will form a Zwitter ionic structure. That means Zwitter means two ions and it will form two ionic structure. Okay, and peptide linkage, as we know that here in the case of amino acid, what happens, we know amino acid is a monomer, right? So it will be bonded with another amino acid and it will also form protein by let me write this down. Okay, so these two are the different, or it can be one and two. Let me take two amino acid. These two are totally different, or it can be similar amino acid. Okay, so here what happened, as we know, these are the monomers, and when they get attached with one or more than one, it will form a polymer. This polymer is being called as protein. So what happened here, the H2O, this H2O will remove from this system and it will form a compound like NH2 R CH C double bond O NH CH R dash and COOH. So it will form this kind of polymeric structure. Okay, not a polymeric, dimeric structure, because here the two amino acids is being combined. Okay, and here in this this is happened due to the this bond is being formed. This is being called as polypeptide bond. Here the CONH part is being called as polypeptide bond, and this chain is being called as polypeptide chain. Okay, so after removal of H2O from the system of two amino acid, it will form a bond like CONH, and this bond is being called as polypeptide bond, and it is highly important as well. Okay, and there is the structure of this is being called as polypeptide bond or peptide linkage, and this is being I have described in the structure as well. Okay, and the protein structure is being important, so please go through that as well. It is not that hard. Of 1 degree, 2 degree and 3 degree, that is tertiary structure of protein, and structure 1 degree structure are mainly the polymeric structure, and the sequence of amino acids is being called as 1 degree, and in the secondary structure there are alpha and beta helix are being found in the secondary structure, and third tertiary structure, this folding of alpha and beta helix will go further and it will rise to macro to major molecular shape, that is fibrous and globular, and this fibrous and globular proteins are being described as here. Okay, and so this is being called as the 1 degree, 2 degree and 3 degree structure, this primary, secondary structure and tertiary structure of protein. Okay, and denaturation of protein, what happened, we know the protein has a structure of helix like structure. Okay, but in the certain conditions like higher pH range or at higher temperature or in certain concentration, this helix structure will be not there. It will be protein lose its biological activity, and the bonds between the helix structure is due to the hydrogen bonding between the bonds and the hydrogen bonding between the elements. Okay, so if at the higher temperature, higher pH, what happened, the pH change is going on, then what happens, this hydrogen bonding will not be there, and due to this the globules unfold and helix get uncoiled and proteins loses its helix structure. Okay, and that is why, because of protein losing its helix structure, it will lose some biological activity as well, and this is being called as denaturation. Okay, and we can see this denaturation in the coagulation of egg when we boil it. Okay, when we boil, the coagulation happens in the case of egg. This is an example of denaturation as well. So in the higher temperature or higher pH, or if we change the concentration, the protein will get unfolded and helix will get uncoiled, and that is why it is being losing in biological activity, and this occurrence is being called as denaturation. Okay, and the fat soluble vitamin, water soluble, this vitamin part is not in our syllabus, so you don't need to know that. Okay, and as you know, the DNA and RNA, the ribonucleic acid and deoxyribonucleic acid. So what happens there and here as well, this is the components of them. And nucleoside and nucleotide, this defined is very much important. Nucleoside is sugar plus base. Okay, and in the case of nucleotide, this is a phosphate. This phosphate PO4 minus is being present. If this ion is present, then it will be called as nucleotide. If this ion is missing, then it will be called as nucleoside. So sugar plus base is nucleoside. Sugar plus base plus phosphate is being called as nucleotide. Okay, and the linkage between the two nucleotides is being called as phosphodiester. Okay, so this is also very important. And functions of nucleic acid, as you know, you can already learn it from the biological aspect as well. The DNA reserves the genetic information, maintain the identity of different species at all as well. Okay, and difference between the DNA and RNA, it can also be important for your syllabus as well, for your exam as well, where this you can see pentose sugar, here is pentose sugar, but this is deoxyribose, this is only ribose. The phosphoric acid is there in both the reactions, but here the nitrogen base are defined, here it is defined, it is adenine, guanine, cytosine, thymine, and here in case of RNA, in place of thymine, in case of RNA we have uracil. So adenine, guanine, cytosine, and uracil was there. So mainly two differences, this RNA having the nitrogen base is different, that is uracil, and in case of DNA it is thymine, and both of them as well as pentose, but here the sugar is for DNA is deoxyribose, and for RNA it is mainly ribose. Okay, so these are the short notes of the biomolecules part, and I have explained it all the way from top to bottom, and I have explained the structure as well. So I hope you have understood this part very well. So thank you very much. Bye.